about the protocol as well as the concepts, key concepts in qPCR for absolute quantification. Okay, so we use the word absolute quantitation of microbial genes in environmental samples using qPCR. I think Dr. Kumar gave you an overview and a nice perspective on PCR and qPCR 2 days back. And we all have some ideas about PCR reactions. Okay, so we know that like if you have one template, if you increase the number of the amplification process to 2 times, you will get about 4 copies. And by the same multiplication process in exponential rate, if you go up to 8 cycles, you are going to 3 cycles, you are going to get 8 copies. Okay, so if you increase it to 30 cycles, you are going to have about 10 to the power 9 copies. So this is well known to us, or rather this is what we expect from PCR reactions. But of course, there are other problems. One of these important issue about PCR reaction is like the use of DNA polymerases. He has also given you an introduction about like why this particular molecule has been regarded as the molecule of the year in 1989. Okay, so earlier to this, prior to the use of Taq polymerase, they were using fragment of DNA polymerase from E. coli, and which had problems. And then because the temperature cannot be raised to a higher level, so this particular enzyme, which was isolated from Thermus aquaticus, is now about a several $billion business. The enzyme from this particular organism is actually marketed in several PCR reactions and others. The point is like so we have what is actually made easy for us, in the sense like you are able to bring the Taq polymerase, and also you are able to bring automation into PCR reaction. Otherwise, what they have been doing is they were actually putting the reaction in each water bath. Every time they have to remove the reaction and then immediately go to the next level when you want to alter the temperature. So those things have been discontinued, so they are able to bring the automation. That made the job very easy for us. Once we have PCR reactions like PCR machines with this thermocyclers, we are able to quantify, or rather amplify the templates to the level which we want. We are able to have good amount of study materials for all our analytical purposes. So amplification made things very easy. So that's the reason like we have been started doing with the several other methods and techniques using these amplicons. And well, that's the reason like we have been focusing more on DNA rather than on other biomolecules like protein and lipids. We are not able to amplify them the way we want to have materials, the way we have with the DNA. So I used to joke with my students, if someone comes out with a procedure or methods for easy amplification of protein molecule or other biomolecules, well, you may deserve a Nobel Prize. So that's the reason like this technology or method have become so popular among all the biologists. So we don't want to stop with one concept like PCR alone, because now there are several methods available with the PCR alone. So we have hot start, touchdown, nested, and then fast, direct PCR, multiplex PCR, and also we have long PCR, GC rich PCR, inverse PCR, and quantitative PCR. So each one has got several applications, or rather specific applications for which you can use a PCR. So the idea of introducing you this particular concept is that you don't limit your applications or imagination only with PCR reactions. So there are several other ways of doing and then gaining information from your PCR reaction itself. And well, I think you also may be interested to know more about direct PCR, because we don't have to even extract the DNA from the sample. Directly you can use the tissue or the environmental sample, and then you can amplify the DNA product. That is also possible in some cases. So we are talking about real time PCR or qPCR. We talk about the ability to monitor the progress of PCR as it occurs in real time. So in the case of normal PCR, amplification is done separately and detection is done in a separate manner. So we have to bring it to the I mean like electrophoretic unit, and then you are able to deduct them whether yes or no, whether products are amplified, templates are amplified, whether we have amplicons in our reaction. So here we are able to monitor the progress of the PCR itself, like how as it occurs. So again, and Dr. Kumar gave you two chemistries. One is SYBR 1 chemistry, and the other one is Tachman probe. So he also explained why we use the term probe, but not the primer in case of Tachman. Okay. So we talk about, like here we will see see about SYBR, and it's a highly specific double-stranded DNA binding dye to deduct PCR product as it accumulates during PCR cycles. Where do they bind? So we know about ethidium bromide. So okay. So they are not only they bind with the minor groove, minor groove of DNA strands, but they are also able to bind as an intercalatase. So, but they are not actually like one molecule of SYBR will bind with only one DNA strand. No, not that way. They may bind it. That's the reason like we have fluorescent intensity varying. There are several reasons for you should know about the fluorescent chemistry if you want to understand like how much SYBR is actually able to bind with a double-stranded DNA. So well, it's and then what is the requirements for DNA binding dye for a real-time detection of PCR? So there should be an increased fluorescence when bound to a double-stranded DNA, and there is no inhibition of PCR. If you introduce a chemical molecule like SYBR, it should not act like a inhibitor for the PCR reaction itself. So it's a big, like you have to take care about like what you are introducing as a fluorescent chemical molecule in the reaction. Well, there are several applications for SYBR. So if you look at them, like they are actually recommended for mass screening, microarray validation, multiple target genes in a few samples, but they are not used for like they are not recommended for SNP deduction, allelic discrimination, but of course you can use it for pathogen detection and viral like gene expression analysis. Yes, you can also use it, recommended to some extent, but not but if you want to determine the gene copy or endpoint genotyping, you need not use SYBR. So there are a couple of issues about use of SYBR. So we should also know where you are able to apply SYBR chemistry or absolute quantitation using real-time PCR. And there are a couple of terms like well, the advantage of using SYBR, which I said to you, like it's you are able to use for monitoring the amplification of any double-stranded DNA sequence. So no probe is required. So the cost is very less compared to your Tachman probe. And the disadvantage is it may generate false positive signals. So you need to be really careful about like what are you generating. And there is an amount of non-specific binding with the SYBR. So there are few couple of disadvantages using SYBR that actually makes this this particular chemistry limited for some of the application which I showed you earlier. Well, what do you mean by quantitation assay? Like the word is something which is new. Like for some of us, it's something it's new in the sense like we don't call it as quantification. We use a word quantitation, but actually it measures the amount of nucleic acid target during each amplification cycle of the PCR, and you can the target may be DNA, copy DNA, and RNA. So there are several ways you can quantify them using real-time PCR and using SYBR chemistry. And couple of terms which have been introduced. Like I think I've seen it in your write-up also. You are given about amplicon and amplification plot, baseline, and threshold cycle. So here I just want to take a minute to explain these things because you need to have an idea like how do we use this concept like for quantification. So when you actually have a reaction curve with the SYBR, so you won't be able to see much of the fluorescence because that's all within the baseline level. So that means like fluorescence signal is just at the baseline level till certain level of amplification could happen. Then only these molecules go and then bind to the double-stranded DNA. Then the fluorescence level will increase. So that's like when the fluorescence level when it increases, we call them as threshold cycle. You put C first and then T as a subscript, like CT we call it. Some of the companies, they also use quantification cycle CQ. So that also I will introduce you. So actually when you talk about threshold cycle at CT value, it's a fractional cycle number at which the fluorescence passes the fixed threshold NTC. That means like no template control. So it's like theoretically you should have a no template control where the baseline signal is actually taken as the threshold level, and beyond which like you are able to see the fluorescence signal. So this is very important, right? And then of course, like bacterial growth curve, we also have different phases of the amplification. So that's what we call this amplification plot, and this is actually the baseline region where exponential phase will start, and of course this is also considered within the exponential phase. And then we have linear and then plateau. This is a non-exponential phase. There are each brand has got a different way of calling the amplification plot itself, right? So what we are now interested is like we want to know about the threshold cycle. If you have an early CT value, that means low CT value, that means you have a good number of templates in your reaction mixture or reaction cup. So that's why you are able to get the sufficient fluorescence signal to get a low CT value, right? So we also use the word early CT. And then if you have a high CT value or late CT value, that means like the fluorescence signal has not come to a threshold level since the templates are not sufficient enough. So we have to, like the cycle takes a longer time to amplify, get the fluorescence signal, then they will reach the threshold limit, right? So this is the two things which you should be very careful, because low value doesn't give you an idea that okay, less number of templates, or high value the other way, right? So this is the one fundamental issue which you should be you should take note of it. And of course, I also introduced you a term like quantification cycle CQ. That's also a crossing point. Some of the companies like Roche, they use the word CQ value, right? Whereas CT is being used by Bio-Rad. And then quantification cycle is actually defined as the cycle at which the fluorescence of a sample crosses the threshold line or the first increases above the baseline fluorescence. So that's CQ and CT. So now you understand, right? And to be useful of a measure of quantity, CQ of a sample must be related to either another sample, and within the relative quantification you can do with that, or you have CQ values of certain independent standards. So from which you can prepare a standard curve, and then you can refer to that like the way you normally do with any other reaction, like what you call spectroscopy reaction. Okay. So the same way you can prepare a standard curve. From there you can know like what is the copy number for an absolute quantifications. Okay. So I will so as CQ alone is not a measure of quantity, which is not a meaningful one, because it only says about which cycle it is actually reaching the threshold level. That's all. Well, and then this is the difference between absolute and relative quantitation. So if you look at the way like in absolute quantitation, so if you want to talk about that, when you have a template 10 to the power 7, if you have a high number of template copies within the reaction mixture, so you will have a CQ value or CT value, whatever you call it now, will be formed early, like within 5 cycles you are able to get the threshold level. Okay. But if you have a 10 to the power 1, only one template or 10 templates are available, and then you need to have at least 20 cycles to go to get the sufficient amplicons in your reaction cup to reach the fluorescent signal for the equipment to detect, for your vision to detect. So with these two so that's the reason like we are making use of this information for calculating the copy numbers, absolute quantitation of your template. And the relative, well, we have 2 genes. So one internal gene is taken as a reference. So that threshold value will actually is also made use of. I'm not getting into it now, because we will now only learn more about absolute quantitation, right? So, but there are theoretically it looks very interesting, because you understand like everything is going very perfect and you have PCR efficiency is good. But within the 20 microliter in your reaction curve, we have a lot of limitations. One is you may not have sufficient number of DNTPs, or you may not have the enzyme, or sometimes you may run out of it, or the reaction may run out of it, or you may have some kind of inhibitors which are present in the cup. So, that will also will reduce the, theoretically we expect 100% efficiency in PCR reaction, but just because of those problems like primer dimer formation, mismatch, and template quality is not good. So, you have problem with the efficiency. So, efficiency will go down. If you look at the way like the baseline reaction, if you put it in a log value of fluorescence against a cycle copy number, the efficiency may it will change. So, that is what it is shown here. Initially it may be 100, more than 100%, then it slowly comes down to 94, 96, 93 and 84. So, efficiency goes down. So, that you should take care of it. So, that is why we prepare an efficiency curve, like what we call is like you prepare a dilution standard curve and look at the slope and see whether the slope is having some, what is the efficiency it shows. There is a equation E is equal to 10 to the power minus 1 divided by minus 3.330 will show you what is the efficiency of your reaction. So, when you prepare a standard curve, you know about the slope, how to calculate the slope, and slope will give you an idea what is the efficiency of PCR amplification. So, these two has to be taken care, preparing standard curve, looking at the efficiency. These two are very important when you want to calculate, like if you want to calculate the number of gene copies, which copy number, I will say copy number gene copies has got some kind of a contradiction. When you talk about copy number, geneticists they are not happy with us because they say you are talking about something else. But as microbiologists and pathologists, we know what we talk about, like we talk about the gene copies. Okay. So, I will there are two ways of looking at the efficiency. Efficiency could be calculated using the fluorescence increase, okay, and the increase in fluorescence, that is efficiency Fi, or you can also prepare a series of dilution series and then calculate the efficiency. So, that is EDS. So, there are two ways. The point is I want you to read this paper by Brackenstock in Applied Environmental Microbiology, published in 2012. So, he has given you us like how to calculate the efficiency in two different methods. Okay. The point is like although the equipment is very sturdy, robust and easy to use, but if you do not understand the basic concept, like the ideas which have gone into the quantification protocols or methods, and you may not be able to use them to your advantage. So, the best thing you should know what are the ways one can calculate the efficiency. So, if if you put the threshold fluorescence in the logarithmic scale against the cycle, then you have a slope that could be also used to quantify efficiency. And if you have a series of dilution and you have CT value and the log number, so here also you will be able to find the efficiency. There are two ways to quantify the efficiency. And again, then how do we prepare a standard curve? So, all of us must be knowing now that like we have gone through several practicals, got an idea like what is the template, what is the length of PCR amplicon, and then if you know the sequence of your amplicon, you are very you are in a very safe place, because you will be able to even know what is the molecular mass of the amplicon. For each one of molecule, you will be able to quantify them. So, make use of that information to know like what is you quantify the amount of DNA in your PCR amplicon in a PCR reaction, and then multiply with the several like Avogadro number constant, you will be able to find out the number of copies of that amplicon present in your reaction vessel rather. So, that is also easy. So, then of course, okay, I am not getting into all this detail in a very short time, but you have to appreciate like these are being taken care of before you get the final value in your software of the equipment, right. These are being taken care of, but you should also know like how they are being taken care of. So, there are several assay, I will say you need to be very careful about like when you set up a PCR, real time PCR assay. Of course, everyone knows about the DNA preparation. If your DNA material is not pure enough, then your whole efficiency of amplification will also get altered, because you don't get the 100% which we set for each amplification cycle. I am talking about each amplification cycle. So, overall is also important, but each amplification cycle also is actually being determined by the DNA preparation. Well, and then if you prepare the DNA, you again go for quantification. Like I will start with the sample which you are using for preparing the DNA itself. Weigh that sample quantitatively, and then you use the same quantity, whatever you quantified material for preparing the DNA. Start from there. That is the material you have to begin with for qPCR. Okay, and then well, quantify the DNA. Then the type of the template, which of course we may not have a control over it, because what is the gene which you want to quantify, that is very important. So, the GC, the repeats, GC rich regions, all will have a say on the PCR amplification efficiency. And then of course, primer design, well, you need to be really careful. I used to ask my students to run the normal PCR, see the products couple of times to see you don't have false amplification, or you don't have multiple bands coming up in your electrophoretic gel, and make sure that your design and then amplicons are good. And then again, like DNTPs, magnesium concentration, Taq polymerase, and internal reference dye by the instrument type. Which instrument you are using, what is the internal standard they want you to use. Sometimes you have to use different chemistry like rocks as an internal standard, so that your baseline fluorescence is actually normalized, and you know what is the starting point from which like you will have a SYBR green fluorescence is measured. And of course, the instrument also is important. If you start an experiment with one instrument, do not change the instrument, don't go to another instrument and try to use it, because although we feel everything is qPCR, sometimes your brand instrument to instrument variations are also will have a say on your data which you are collecting, and the kind of controls which you set, NTC and buffer. Of course, data analysis, melting curve. Nowadays this is available in the software itself, so you can have an idea whether you are only getting amplicons or some other dimers is also appearing in the melting curve. These analyses have to be done. Well, couple of run you have to do without actually going for the actual quantification of your gene, and then start your experiment and prepare your standard curve. There are several ways of preparing the standard curve. So, when this is the important step, because if your standard curve is going to determine, or rather it is going to play a major role in the values which you are going to get. So, if you are using a, I'll say if you actually amplify a template, a gene of interest, and then purify it, make serial dilution, and then you can go for a standard curve, or you take the amplicon, clone it, put it in a vector, isolate the vector, purify it, prepare serial dilution, and then you go for quantification. So, the level of precise information which you want, you can design, and then you go for the preparation of standard curves. Of course, then the kind of material which you are using for quantitation is also important. Suppose you isolate you wanted to quantify the material in a particular soil type, you try to use the same soil for preparing standard curve. So, you use one soil type and then try to use it in another soil type, extrapolate them. We have some problem. We will have some problem because inhibitors usually will come up. So, even after you prepare the DNA, you purify them, get a good template, and then you go for PCR amplification. Avoid all those impurities after DNA preparation. These are some steps which you have to introduce when you want to have a high quality data. Well, this is the way you have to prepare a standard curve, log DNA dilution, and then CT value. So, you can also get the regression equation, which actually should say more about what you call R square value should be more than 0.99. Then you will be able to use it. And then if you look at the slope, well, you will be able to understand about the efficiency of the reaction, and that can also be, you should also make use of them. And of course, as I said to you earlier, you should be able to quantify the number of copies present in each amplicon which you are going to use for your standard curve. So all those details I have given it in this. And then the protocol has to be prepared for each gene of interest, and say use it carefully for each gene which you are going to put it for your qPCR analysis. Well, I think this you can pick it up from any of the website. Thermo Fisher has got a nicely detailed the protocols in their website, modified as you want to do it in your laboratory, and go for quantification, right? And of course, you may have problem with the no PCR product. You can avoid it once you have a normal PCR done, and you know your PCR products are coming for the primers which you have designed. Also, I'll say that as far as qPCR is concerned, do not go for amplicon length more than 250. Try to have within 150 to 250, because if you have a longer amplicon length, that is going to affect your quantification. Like efficiency is also getting affected. So you select a primers set in such a way that your amplicon length is not crossing, like it's within 150 to 250. That's from our experience. So if you do have it, well, your slope is going to get problems. And then look for primer dimer, and then also check whether multiple bands are gel or multiple peaks in the melting curve. Avoid all of them. And then non-specific amplicon also you should avoid. Well, you are set. You can now go for your qPCR. You can ask me questions related to that. Although my equipment is very, like the one which you used, we can use for absolute quantitation. But understand, there are new methods of looking at the data, or quantifying the number of gene copies which are present in the environmental sample. So you improve them regularly rather than sticking out with only one method. Several new ways of statistical analysis is also coming up, and of course you should have more replications to avoid the failure, or to improve your accuracy in the data, improve the precision. Any questions now? And for this particular lab practical, I requested one of my students, Ilekia, to bring her own DNA sample and PCR SYBR green primers. So setting up experiment is going to be easy because it doesn't involve much work, because everything has been done by her. So she has brought DNA sample and primer set and SYBR green strips. So all you have to do is simply add it in the master, and use the master mix and set the reaction. She will show how to prepare, set the equipment for quantification, and also to collect the data and then analyze the data. Important point is, before you select, you work with any particular gene, prepare the standard curve so that you are very sure that you will be able to count, or rather quantify the gene copies. Right. Okay.
Okay. So that was a lecture about absolute quantitation. This lecture is equal to the lecture what I delivered here on real-time PCR. So with that, I think both the technology, that is relative quantitation and absolute quantitation, that is communicated to you. Now you go back to the lab, you do this practical absolute quantitation. Once the reaction is set up, then we will start another very small practical about the data analysis of relative quantitation real-time PCR. Okay.